right, it is 12:01, so let's get started. I have one more person who will be making up the exam, so I don't have them for you, but I posted the grades. In general, the average was higher than the other exams, but it was a pretty wide range. So I'll give those, well, you guys will be on Zoom, but you guys on Zoom will see them on Monday, and then if you guys want to come by my office and see them before the following week, you can. But we'll take a look at those, and after this, you have one more like regular exam, which will be the very last day of class. I think that's when I have it scheduled. And then, unless I put a review day in between that and the final, I can't remember. But one more regular exam on this material that I'm about to go over, and then the final is cumulative. So there won't be any new material on the final. You'll have been tested on it all at that point already. But just a heads up for what's coming up. So still 2 more exams. The final is worth the same as the other exam, so it's not worth any more. It'll just be longer because it's cumulative. Any questions about that? No?
What is our final?
Oh gosh, May 7th, I think. Yeah.
Yes.
Yeah, okay. So yeah, your final is May 7th. I think it's at 1:10 in the afternoon. So I'll talk about that more. I think we'll probably, yeah, we'll do it over in the labs, just like we do the regular exams. All right. So this is the last unit of new material. You guys are so close to the end of the semester. You have made it very far. We're still talking, we're kind of continuing on with DNA coding for all the proteins that make up our bodies. So we're kind of doing the next step. We talked about DNA replication, how it replicates itself and makes more of itself for all the new cells that have to be made. Now we're going to continue on and talk about how the proteins are actually made from that DNA. So that'll be the next couple of chapters that we're talking, or at least the next couple of lectures that we'll be covering in here. Before I forget, and while everyone's still actually paying attention, there is a worksheet on transcription, which is what I'll get into today, that's due on Monday. So I'll see how far we get. But as of now, there's a worksheet on Canvas called Transcription Worksheet. It'll be due Monday. Just upload it like normal to Canvas, and then we'll go over it. But it is for a grade. It's for, you know, one of those miscellaneous assignments. So make sure you do that, assuming we get far enough today. So if not, I'll let you know, but it's due Monday as of now. Okay, so let's kind of step back. I guess I kind of already talked about where we've been and where we're going. So by now, where we've been as far as this molecular stuff goes, so at the molecular level, you know how DNA is structured. We talked about the double-stranded aspect and then this base pairing. Base pairing we're going to continue talking about through the rest of this whole process of making proteins. In DNA, A paired with T and C paired with G, right? When you had those 2 strands coming together, those were the base pairing rules. And yeah, we looked at the whole process of how new DNA is made. You learned all of the enzymes, the proteins that are involved in that process, kind of the process from start to finish. All right, so you know all about DNA. Now we're going to use that DNA essentially to make RNA. That RNA is going to be used to make proteins. That's our ultimate goal. So you guys remember me talking about the central dogma of biology, hopefully. DNA is going to code for RNA, which is going to code for protein. So these are all just codes based on those A's, T's, C's, and G's. That's what our whole body is based off of, which is kind of crazy. So yeah, coding for RNA, which is going to code for proteins. The process of making RNA from DNA, so basically reading that DNA to make this, I'll talk about what RNA is. That's called transcription. That's what we'll talk about today. So DNA is being transcribed. So if you transcribe something, you're basically writing it from one version to another. So that's going to make the RNA, that's the new version. And then that RNA gets translated. So transcription and then translation, that's the order of what's happening here. So DNA to RNA is transcription, RNA to protein is translation. Those are in alphabetical order if you look at them. So transcription comes before translation if you were putting these in alphabetical order. So that's hopefully a helpful way to remember the order that they occur in. So today we're doing transcription. Next week we'll get into translation. Okay, so I want to talk first a little bit about genes and gene structure before we get into how this DNA is read and then turned into an RNA molecule. So a gene, as we think of it, we talked about it a little bit when we were talking about obviously Mendelian genetics and how traits are passed along. From a more molecular perspective, when we're looking at a strand or a molecule of DNA, a gene is just a region on that DNA. So you can think of it as a long segment of the DNA. That's going to be a gene. It contains a coding sequence for a very particular protein. So those base pairs in there's all these base pairs that are holding this DNA double helix together. Those letters are very specific and they will code for a very specific protein that will be made from that gene. Contains, yeah, so that sequence of letters will be used to make the RNA, like I said. That's our first step, transcription. On a gene, you typically have 3 regions. There's only one of those regions that actually codes for the protein. That's going to be the middle, the coding region. In front of that, if you're kind of reading from left to right, you can imagine we're reading this gene. You have the promoter, which is just kind of a place to indicate to the enzyme that's going to make this RNA molecule, this is where you need to start. At this point in the coding region, this is where those letters become really important, A's, T's, C's, and G's. So that's our coding region. And then at the end, there's a termination sequence, which tells that protein that's making the RNA strand to stop. So stop reading this code and then break off and you're done making the RNA. So promoter, coding region, and then a termination sequence. And I'll talk a lot more about this region, but I'll talk a little bit more about the promoter and the termination sequence in a minute. So that's the general structure of a gene. An overview of protein synthesis. So this is hopefully kind of helpful. Here's the central dogma in sort of a simple diagram. So DNA to RNA is going to be transcription. DNA is double-stranded, RNA is single-stranded. I'll talk more about that in a minute.
So it's like you split the DNA in half?
Kind of, yeah. Yeah, that's a good way to think about it. It's a totally new molecule. It's not like you actually take half, but yeah. And then reading that RNA, so the code, these are going to be those nitrogenous bases again, the A's, the T's. Well, in RNA, there's one difference, but you know, the same letters essentially. That'll be read to make a protein, which is just a bunch of amino acids strung together. That's going to be translation, RNA to protein. Where this is all happening is, so transcription is happening in the nucleus. If the RNA is being made from the DNA, we have to be in the nucleus because the DNA is in the nucleus. So this has to happen in the nucleus. The RNA then leaves the nucleus, and the translation part, making the protein, happens out in the cytoplasm, just not in the nucleus. I'll talk about mRNA in more detail, but this is essentially the mRNA is a messenger RNA. It takes the message from DNA and makes the protein. So, but I'll get into that later. I don't know why I have that defined here. It'll be defined again later. All right, so that is a general overview of protein synthesis. Like if we zoomed way out, this is what's happening. We're taking DNA, we're making an RNA molecule. That RNA molecule is leaving the nucleus, and we're stringing together a whole bunch of amino acids to make a protein. Okay, stages of transcription kind of line up with the regions on a gene. So hopefully you recognize these stages: initiation, elongation, and termination. Those were the three main stages in DNA replication as well. It has to get initiated, the strand that's being made gets elongated, and then it ends at some point. So these are just three general stages that apply to the DNA replication that we talked about, and then it'll apply to transcription and translation as well. Okay. Initiation happens in the promoter region. This is where it's kind of a signal to get the process started. Elongation happens when the protein gets to the coding region as it's reading kind of from left to right. Termination obviously happens in the termination sequence. Okay, so let's go through each of these. It'll feel kind of similar to DNA replication. I'm going to talk about complementary base pairing. We're basically making, like you said, a simplified version of DNA, just a one-stranded DNA. There's a few differences that I'll talk about. So the initiation process, how does this all get started? First, that promoter region has to be detected. So the detection of the promoter region by, I'll talk about this in a minute, but RNA polymerase. Do you guys remember what the really important enzyme in DNA replication was? The one that like adds the new bases? It's really similar to this.
Just DNA polymerase?
Yeah, exactly, DNA polymerase. So DNA polymerase, like I said, if you remember any of the enzymes involved in DNA replication, remember that one. RNA polymerase is pretty much, thankfully, the only protein or enzyme you have to know for this process. It's kind of the workhorse. It does everything. DNA replication had all of those others, the ligase and the topoisomerase and helicase. In transcription, RNA polymerase is pretty much it. It's doing everything. So the RNA polymerase is what has to recognize basically this promoter sequence. So it's going to find that promoter sequence and latch on. The promoter sequence usually starts with or has in it a bunch of thymines and adenines. So they call it a Tata box. So lots of T's and lots of A's strung together. It's typically the indicator for the promoter region of a gene. Okay, and then here's RNA polymerase coming in. It recognized it, so RNA polymerase is coming in and binding to that promoter region. Like I said, that's the main protein in transcription. I don't think I even have any others listed in here. So RNA polymerase is it, thankfully. And as you can see in the bottom there, oh, sorry, you can't see RNA polymerase on this screen. That's annoying. So you guys on Zoom can probably see the RNA polymerase. It's kind of showing up as this big blob. So just imagine a big enzyme here that is RNA polymerase. It's unzipping this region of the promoter. So the DNA, like I said, double-stranded, so you have to open it up in order to read that code, right? So RNA polymerase is going to unzip the DNA.
Is this like the thing that was happening in the Movie Sister video when they were like unzipping and then...
Yeah, with DNA replication.
Yes.
Yep, yeah, same idea. It just gets unzipped by RNA polymerase this time. Yep, exactly. Okay, so that is the initiation process. We've got essentially RNA polymerase finding the promoter region, binding to it, and then unzipping the double helix to open it up and expose the genetic code in here. Okay. I think that's all of initiation. Oh, a little bit more, sorry. In the initiation process, we include the first attachment of the first RNA nucleotide. So the nucleotides are those ATCG. There's one difference, like I said, like I mentioned in RNA. So one of those letters changes, which is kind of annoying. I'll talk about that in just a second. The attachment of the first nucleotide. So to that unzipped DNA, it's going to be complementary base pairing again. That's done by RNA polymerase. Transcription factors are going to help to stabilize that unzipped DNA. So that is, I guess I do have another protein listed in there, but they just stabilize it, transcription factors. There's a number of them, but we don't need to go into detail on that. They just help to stabilize it, keep it unzipped. Okay. So that's the end of initiation, I think that's all I have. Yeah, okay. So moving on to elongation. So this is where we're actually really making the RNA strand. We're synthesizing or creating the RNA strand. Just like we created the DNA strands, it's going to be done in the 3' direction. So we still have that wonderful 5' to 3' idea, but new bases are going to be added to this RNA strand on the 3' end, just like before with the DNA. RNA polymerase is doing that. So RNA polymerase is bringing in these nucleotides. So the pink here is going to be the RNA transcript that's growing. The blue, or on the screen green, this is the DNA, right? So the DNA is what's being read, but just this strand is being read. So because RNA is single-stranded, it only needs to read one side of the DNA. So the RNA polymerase is reading these letters on the DNA. So for you guys on Zoom, reading these letters on the DNA and bringing in the correct complementary bases. You'll notice a U. That's the one that's different in RNA, and I have that here in just a second. So RNA polymerase is bringing in these new bases, matching them up with the complementary DNA bases.
Oh, this is like the prime, okay.
Mm-hmm. All right, so the nitrogenous bases in RNA, or nucleotides, whichever you want to call them, think of them as. We still have adenine, guanine, and cytosine, but to complicate things, instead of thymine, we have uracil. So there is no T, you're never going to find a T on an RNA strand. There's a U where there would be a T if we were talking about DNA. So the base pairing C and G still go together. Obviously now A doesn't have a T to go with, so it goes with U. So AUCG. On the DNA strand, obviously you'll still see T's, right? So you still have that base pairing. So if there's a T on the DNA, exactly, it'll still pair with an A because there is an A on the RNA. The only difference when you have an A on the DNA strand, you bring in a U instead because there is no T. It gets confusing. It's just easy to kind of get a little bit turned around, because right here we have T's and U's, which gets confusing. So you have to think about, okay, am I looking at the DNA strand? If so, there are T's, what would that pair with? Kind of slow down and take it step by step. And just remember, if you're drawing out or writing out a sequence, which I'm gonna have you do on that transcription worksheet, the sequence of complementary bases for the RNA strand, never put a T. If you want to put a T, think, no, I have to put a U. So for you guys on Zoom, so we have T's on the DNA, right? So we can pair that with an A that's still present in RNA. But here's where you have the change. You have an A in the DNA. Normally, if we were just talking about DNA, there would be a T here, but RNA has uracil instead of thymine. So you have to pair it with a U. That is very easy to confuse, and students do it all the time. So try to keep that straight in your head to the best of your ability. Again, just kind of slow down and think about it. That's my best. I have to slow down and think about it half the time when I'm writing out the complementary bases. All right, so that is the elongation process. I don't know if I have any more information on the next slide. But the whole idea is bringing in these new bases, remember, adding to the 3' end, and building this single-stranded RNA. So this is what we're building, the newly made RNA. That is the whole goal of transcription. Oh yeah, and you'll notice I might have this elsewhere. The strand on the DNA that's being read, because I said we only have to read 1/2 of it, right? That's called the template strand. That's down here. I think I might have it defined elsewhere. Oh yeah, right there. Template strand. So the template strand is what's being read in order to create the RNA. Nothing's happening up here. It's not being read.
So only the bottom ones are getting read.
It depends on what gene needs to be made. So sometimes it'll be the top. Yeah, but there's only one. Only one of them will be read at a time. So this is just another image of the elongation process, kind of a simplified version, I guess. You have the RNA polymerase, which again, for some reason is not showing up on the projector. You have the template strand here being read. Again, yes, it's on the bottom, but that's just, it can be the top. You have the template strand here. The other strand is called the coding strand, but the template strand is what's read. And then you have the matching up of the RNA nucleotides with the DNA. So you read the template strand, you have a T on the DNA, you add an A on the RNA. If you have an A on the DNA, remember U goes on the RNA.
So the template strand might still consider, okay, you just split in DNA and then you put the RNA.
And you ignore the coding strand. Basically, it's called the coding strand because this code is the same as what the RNA is because they're both complementary to this, right? Except in DNA, you have the T.
Yeah.
So the template strand is what's being read. It's the template for the new RNA. Okay, we're already to termination. We're flying through this. So the termination process occurs at the terminator or termination sequence on the DNA template strand. So we talked about that. It's after that coding region. And here you can see the RNA, we call it the RNA transcript, being released. So here it's being made, we're still kind of elongating. You get to that termination sequence, which is just a specific sequence of nucleotides that you don't really need to worry about. It just tells the whole process to stop. And then the RNA strand or the RNA transcript, the RNA molecule, whatever you want to call it, breaks off. The DNA rezips, well, it's rezipping the whole time, but it comes back together. It's not damaged at all. It can be reread again and again and again. And then, yeah, again, you can't see the RNA polymerase. Anyway, there's an enzyme here, that RNA polymerase, that protein coming off and going its own way as well. Right, and then the last thing I have here is just the renaturation, which is the rewinding of the DNA. So it gets wound back up into the double helix. Ah, yes, videos. I haven't watched these videos in a while. I can't remember exactly what they are, but let's watch some transcription videos. So that's the whole process. Now that you know about DNA replication, hopefully, like the base pairing is the biggest thing. So if you get the base pairing idea, hopefully that transcription process was a little bit easier to understand. So yeah, let's see. I know this first video is kind of boring, but it's not like super exciting, but it's helpful. And I don't remember what that YouTube video is.
A structural gene is made up of a sequence of bases in a DNA molecule consisting of a coding region with an upstream promoter and a terminator downstream of the coding region. The transcription of a gene into a messenger RNA sequence is initiated when RNA polymerase attaches to the promoter region and forms an open complex. RNA nucleotides are then polymerized from nucleotide triphosphates to make a messenger RNA.
So some of that, this does like nucleotide triphosphates. You don't need to know what those are. Any of the terms you don't recognize in this video, don't worry about. It's just helpful to kind of see it in action.
The messenger RNA sequence is elongated as the RNA polymerase moves down the DNA molecule until the RNA polymerase reaches the terminator region. Transcription is terminated when sequences in the terminator region are encountered. This causes RNA polymerase to dissociate from the DNA molecule, and the completed transcript is released.
All right, so straightforward, like I said, kind of dry, but it's helpful to see that little bit of action in that animation. Now, let's see what this is. Sure. Oh, okay, yeah.
What you are about.
So this is like a computer model, a really high-tech computer model of how RNA is made from DNA.
C is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. Begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA.
Yeah, the RNA is that yellow coming out.
To make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
Yeah, so it gets to that terminator sequence at the end, and then it just all breaks free at that point. Yep, that RNA polymerase will probably go to another gene and then start doing the same thing. Yep, good question. So yeah, I don't know, it's kind of a cool computer model, I guess. I don't know what the noises are all about, but I don't know if that's how it actually sounds in our house. Somehow I doubt it, but somebody spent a lot of time putting that together. Okay. Okay. All right. Any questions so far? Hopefully this is kind of straightforward. Let's go back to here. Okay. Unfortunately, once we get to the end of like that termination part, well, after termination, sorry. So everything's broken free, the RNA transcript is off on its own. That transcript still has to undergo a few alterations, like a little bit of processing in order to finalize the process. Oops. So at the end, that piece that broke off, basically, that RNA transcript was called pre-mRNA. It just means it's not like a fully mature RNA. So there's a few things that have to happen in order for it to be the final mature RNA that's going to leave the nucleus and go on to be translated into a protein. This is only in eukaryotes. I'm really not focusing on the prokaryotic aspect. That's in bacteria. But yeah, for obvious reasons, I'm going to focus on eukaryotes here, which is us. Okay, so the pre-mRNA needs processing. So this is what we call the post-translation or post-transcriptional processing. A couple of caps have to basically be added to each end. On one end, we call it the 5' cap. It's on the 5' end of the RNA. And basically, it's just a special guanine nucleotide, which is the G in that genetic code. So a special guanine nucleotide is added to the 5' end, so you get this little cap. It's represented here by the blue dot. So you have a 5' cap. At the other end, you have a bunch of adenines added, and they call that a poly A tail. So many A's, many adenines added to the 3' end. What those essentially do is protect the RNA molecule, the really important part that's going to be read to create the protein. It protects that from when the RNA has to exit the nucleus. It's kind of a dangerous journey for the RNA to get from the nucleus out into the cytoplasm. So those caps really help protect it. You can think of them as like little, I don't know, little helmets on the end, little protective caps. And the other thing that needs to happen, so we have those two caps, the 5' cap, the poly A tail, something called splicing occurs. So interestingly, in these RNA transcripts that are created, you see these red and green regions. So here we have red and green. The red ones in this diagram are called exons. The green ones are going to be introns. The introns have to be taken out, and then the exons are basically glued together in order to create the unbroken coding sequence. There's a lot of research and still kind of a lot of debate about what these introns do, considering they're put in and then just have to be taken out. I'm not going to get into all of that, but it's easy to think about the exons as the ones that have to be, as the ones that have to be taken out, right? Like exons exit. The way to remember that introns are the ones that are taken out of the eventual mature RNA is that introns are in the way. So just remember, introns are in the way. I think 50% of people, students in the classes where I cover this exon intron idea, think that introns stay in for obvious reasons. It's kind of poorly, I think they're poorly named. Introns are the ones that are removed. Exons are then glued together, and that's the final mature mRNA molecule. Just a bunch of exons with a 5' cap and a poly A tail. And that's what moves out to the cytoplasm for translation. All right, so here we have, I put in this little box why we have those caps. Just because I don't think I had it on the previous slide. So that's written there for you. Here you can just kind of see a different version of what I just showed you. We have the 5' cap. We still have the exons and introns. So this is not a mature mRNA yet. We have the poly A tail on the 3' end. The exons are what, or sorry, see, I do it. The introns are what needs to be removed. So they'll be spliced out. It's called splicing.
And all the exons would just come together.
Yep, they'll all come together. And I don't really know the process for how that works, but we don't have to worry about it. Okay, and here we have the final mature eukaryotic mRNA. Don't worry about the UTRs here. Just kind of another region that's not translated. You have the main protein coding segment with those couple caps that I had mentioned. These are the, yeah, this is all the exons put together. The introns are already gone.
Who did that?
The UTR is just, so the polyadenylation signal, that just means this is where the many adenines need to be added. Don't worry about it. Don't worry about that part. I'm not going to ask about the polyadenylation signal, but UTR just means an untranslated region. So these aren't used to make the protein. It's really just this red part. Wow, that's it. That's all. That is all I have for today. I didn't think I'd get through all that. So yeah, take some time if you have time now. Start on that transcription worksheet. We'll get into translation on Monday. But do that transcription worksheet. It covers kind of a great review, well, a good review of most of this information. We'll go over the answers on Monday, but definitely turn it into Canvas by the beginning of class on Monday. And that's it.